All righty, everybody. I think we'll go ahead and get started with our next Ivan Roundtable workshop on biomolecular NMR applications. At this point, I'm going to turn it over to David Rice.
Welcome, everyone. And actually, we're switching gears slightly as we're moving our topics towards biomolecular NMR for this session. I'm hoping that we will be doing many more of these and in other types of NMR. John Webb, our sponsor, could not be with us today. And so Eric is going to play a short message from John.
Thank you, Dave, and good morning, everyone. Good afternoon, good evening, wherever you may be. Once again, great subject material today. I've got a very, very good panel lined up and looking forward to it. MR Resources and Q1 Instruments are very happy to be bringing the Ivan meetings to you. And MR Resources, of course, have been around for over 35 years at this point, and providing reconditioned NMR systems, magnets, consoles, probes, service contracts, quench magnet recovery, and certainly moving and relocation. In fact, they've got a very impressive inventory of reconditioned instruments on hand, and three in particular, very spectacular offering, all Brooker Avance 3, 600s, 700s, and they've even got a 950 in inventory at this point, all with cryo probes. Certainly check them out, get in touch if any of these interest you. And Q1 Instruments, a little bit newer to the industry, but Eric, could you give us some brief information on Q1, please?
We invite you to get to know Q1. Q1 manufactures consoles and complete systems from 600 megahertz and below. And we also offer complete automation with upgrades for customers' existing magnets, 300 to 600 as well, with our patented STM probe, 24 or 60 position auto sampler, preamplifier, and 2-channel console. Q1 offers excellent performance at an unbeatable price, and our central feature is our Smart Tune and Match probe, which consists of a robust hybrid tuning mechanism with faster tuning and unmatched reliability and improved throughput. It's a patented design, free from drift and hysteresis, with excellent signal and noise and superior solvent suppression, and made by our Q1 team in Zurich, Switzerland. The probe configurations include inverse probes, as well as probes where you can select fluorine on one or both channels. These probes are also available for Brooker invariant systems using our preamplifier and network analyzer that interfaces with both Topspin and VNMRJ. We've installed these systems on both variant and Brooker systems at the University of Toronto and Dow Chemical, among other places in the world. If you want to know more, please get in touch with us. Thank you very much.
Thanks, Don. So Chris also could not be with us today, so I will take over his role and give you a brief idea of what's upcoming for our research workshops. This December workshop is our last for this year. In January, it's looking like that we'll have Istvan Peltzer from Princeton leading a workshop on metabolomics. And in February, Roberto Gill will talk about dipolar couplings. Then we've got for March, Jose Napolitano talking about QNMR. And we should not forget the ENC in Asilomar again this year. And we will be doing our regular hybrid meeting live from the ENC. This year we will be really live with an audience at the ENC as we were last year on the East Coast. And tentatively for May, we've got something lined up with Gennady Kurich from Merck and talking about water suppression. So we've got a lot coming up for the year. And with that mentioned, I'll turn things over to our panel leader, Rajan Murali, who will be talking about both, who will have people talking about both liquids, biomolecular NMR, and my interest, solids bio-NMR. And so I'll turn things over to Murali.
Thank you, Dave. Good day to all. The thing is, it's a nice opportunity for all of us to get together in this forum and share the wealth of knowledge that we have amongst us. This has been going on very nicely. And recently, Krishnamurti contacted me and asked me to arrange a talk on biomolecular NMR, because so far this forum has been focusing on small molecule applications and methodologies. So Krish wanted to widen the audience and prospects. So he asked for biomolecular NMR applications. And the moment he told me, I got two names popped into my head. One is Ranajeet Ghose, Professor Ghose, who's at CUNY, New York. He's an expert in solution NMR, both theory and practice. And he's focusing on interesting biomolecular applications in protein ligand interactions. And then on the solid state side, I have the privilege of interacting with our in-house expert, Professor Andrew Newcook. And he is pushing the solid state methodology to the frontiers, and it is almost coming to like liquid-like NMR applications in solids with his fancy 100 kilohertz spinning probes. So without much ado, I'll invite Professor Ghose to give his talk first. After he finishes his talk, probably we'll have a 5 or 10 minutes gap to address some of the questions you may ask based on his talk, and then we will move on to Professor Andrew's talk. So I invite Rani to give his presentation.
All right. It gives me immense pleasure to talk to this forum. And one of the really cool things I see is I see an old friend in the audience, without whose help this work would have never happened. Kaushik Datta, who used to be in the neighborhood, maybe a decade ago, or 5 or 6 years ago. And the initial part of this work started with his help. And then after he left for greener pastures, we sort of stopped in it, doing NMR on the system. And then we started doing a lot of computational approaches. And then finally, we could understand some of the NMR data based on the computational work. And so we revived it. And so I want to do a shout out to Kaushik, without whose help this work would have never happened. Anyway, my training was NMR methodology, both experiment and theory. And this was my intent when I started my lab, 2 decades ago. And over time, we sort of moved away from the methodologies, for various reasons. And then slowly transitioned into other approaches. And now my lab does all kinds of things. NMR is it's still a major approach in my lab. I would not say it is the dominant approach anymore. And so we do computation, we do crystallography, we're beginning to do cryo-EM. And so we are more driven towards applications. But I'll give you an example. I'll tell you about some work which initiated many years ago. It was stalled for a while, and then we recently finished it. It's on a very interesting system we've been investigating for some years now, and this is a protein kinase, which is and it's a tyrosine kinase. For those of you who are interested. This is a bacterial tyrosine kinase, and it's sort of I can get into the biology if you guys are interested during the question hour. But basically, this is very unique in a sense. It doesn't look like the tyrosine kinases we have. And so this system we were interested in is to understand how it actually performs its function in various ways, both, in a mechanistic sense on how it actually phosphorylates on tyrosine using ATP, how it is regulated by its phosphatase, which is this low molecular weight tyrosine phosphatase, WZB. So ZC is the kinase, ZB is the phosphatase. Just that's all you have to remember. And the details are unimportant here. And they function. There's a very large medical interest to this because a lot of pathogenic bacteria tend to utilize this mechanism to be infectious, but that's besides the point. Anyway, so this is the system we wanted to focus on, and this can provide sort of a general view of how transient complexes or weak complexes can be studied using a variety of combination of NMR techniques, some of which are relatively common, others which are somewhat not uncommon. Okay, so basically, overall, you can forget about the system. All you have to think about these two interacting protein partners, which interact relatively weakly. And we tried for many years to crystallize the complex, and that didn't work. And so, we decided to go with this sort of approach. So let's first look at the system. So the system consists of a kinase domain, which is cytosolic. So it's a soluble protein. It's got a bunch of these phosphorylatable residues here on this very disordered tail. This is the core domain, which is ordered, and this is a schematic representation of the phosphatase. So what we're going to look at is what we found was basically you can do an ITC measurement, and you find that this tail is not required for the interaction between the kinase and the phosphatase. So this is good for us in the sense that we can sort of get a structure of the system using just the core domain and the phosphatase. Because if you were to do a computational approach, a computational docking approach, which we're going to use, having this tail which is disordered would immensely complicate the computational problem, because you would the sampling problem would be immensely more complicated. Anyway, one of the other things we just to mention is the heat release is very low for this interaction, and so there's a lot of so it's mostly entropically driven, and that sort of complicates doing biophysical measurements using ITC. So, as you will see, we switch to NMR. So the advantage here is there is a crystal structure of this piece of the core domain. There is a crystal, there's an NMR structure of the phosphatase. So this was actually Kaushik. This work was done with Kaushik doing a lot of this work. So I'm glad he's here, so he can see where we got to at the end. Anyway, so we did the most naive thing one does. Well, we took this core domain, we assigned the backbone resonances, and then we added the other domain unlabeled. So this was N15 deuterated, and we added the unlabeled phosphatase, and we saw a bunch of perturbations, mostly line broadenings, right? And so then we saw, well, so this is the interaction area, the orange part which showed the largest amount of spectral perturbations. So then the obvious thing to do is look at the conserved areas, right? We looked at the conserved areas, and we found, and then you make a bunch of mutations, and then you have biological readouts, and you see what happens. And we found that that there is a helix with these three residues which are fully conserved in this family of proteins. These were the ones which are key in interaction. Okay, so that's a problem solved. That's an easy problem, right? What about the other end, right? The other end gets completely messy, because when you add the kinase, when you look at the phosphatase, the interaction partner, and when you add the kinase, what you see is essentially an entire face is completely broadened out, right? So we can't, and if you do try to do the same thing where you actually try to look at conservation and try to figure out what could be interacting, this becomes a problem because these phosphatases tend to be a little bit promiscuous. So the only parts which are actually conserved are the catalytic sites. So something else is doing the interaction, the docking interaction, which cannot be figured out from this sort of naive sort of analysis. So, and as I said, this was published many years ago, maybe, this data, the perturbation data, maybe in 2014 or something, a long time ago. And, we said, well, perhaps the way to go about this interaction would be to try to co-crystallize this. And this is not something we managed to do. Not unexpectedly, because you need high levels of concentration both, and then the interaction is relatively weak, and we just couldn't co-crystallize it. And so we didn't know what was going on. So we decided we're going to take an alternative approach. As I said, there are structures of both of these entities which interact. So we could use some kind of data-driven docking. And how would we do that? The way we would do that is using a set of restraints. And the set of restraints we were looking at were, and I'll get more into this. Static chemical shift perturbations or static spectral perturbations were problematic because it didn't really let us focus in on a surface. And we would still use it, but at least on the WZC-B side, on the phosphatase side, it just wasn't specific enough. So it wouldn't be extremely useful. But in order to get a well-defined ensemble for the complex, you also needed some unambiguous actual distance measurements, and that we were going to do using paramagnetic relaxation enhancement. And this we were going to combine with this very elegant software suite called Haddock, where I'll get a little bit into this in just a bit, where you can combine all of this data and you can do data-driven docking, and then you actually validate your system. So spectral perturbations, as I said, static spectral perturbations were unhelpful on the WZB side, on the phosphatase side. So what about transients spectral perturbation? Now that's something we looked at. So those in this audience know that if you have some kind of conformational exchange, right, systems exchanging between multiple or two conformations, assuming that they are in a sort of the appropriate time scale, you would end up getting line broadening effects. And these line broadening effects can be measured using, NMR relaxation. Depending on the time scale, one use measurement, rotating frame relaxation, R1 rows or T1 rows as they're called, or these series of CPMG type sequences, which attempt to characterize the exchange time scale and characterize, whether the exchange exists or not. So in this case, it's slightly different, right? Where the exchange arises not from conformational change, but essentially exchange between two states, in which one of the states is the free state, the other one is the bound state. The assumption is when binding happens, you're going to see chemical shift changes. And if at low levels of ligand, which is the kinase in this case, you're going to end up seeing chemical shift perturbations. And if you have low levels, it's not a stably bound system. So you see transient changes, and this should in turn cause line broadening like we saw here. And then we can characterize that using a sort of a CPMG type scheme to measure exchange, where the exchange is not due to conformational change. Well, it could be due to conformational change, but the primary effects are essentially due to binding, due to exchange between bound and free state, assuming that you're in the correct regime. And we know that we are in this case. Okay. So here the chemical shift changes. Normally, when you look at conformational dynamics, this this chemical shift change essentially is between well, this is a two-state model, so between two conformations, but in this case, it's between a free and a bound state. And so what we would do then, we would have the free phosphatase, and then we would start adding low levels of the kinase and see which regions sort of light up in terms of exchange. Of course, in the absence in the absence of any of the added ligand, we don't see any exchange, which doesn't mean exchange doesn't exist. It just doesn't exist in this time scale. Anyway, then we add low levels of ligand, and FP is the bound fraction, and then we see these regions light up. And the thinking, you can call it naive, but this is a good first order approximation, is that the contact points are going to be perturbed first. And so, this would give us a more concrete region which appears to interact, take part in protein-protein interactions. And so we can then map it on. As you can see, these build up, and finally, ultimately, when you have saturation, you should reproduce what you saw in the comparing the free and the saturated bound states. Okay, so this in principle could be used as a conformational restraint similar to what you would do for static chemical shift changes. Okay, so there is another way to do quality control here. Okay, so as I said, the relaxation rates which you measure, the effect on line broadening, essentially are a measure of the exchange between the free and bound states. And so if this exchange is on the fast time scale, realize this is not on the chemical shift time scales, but on the time scales of the relaxation rates which are being averaged. So the relaxation rates, the free and bound states. If the exchange, which is what under most concentrations for protein NMR, this essentially means a K-off, and we can calculate a K-off from this. And we've done SPR measurements earlier, so we can get a K-off. And in this regime, essentially, and in the limit of extremely fast pulsing, you should get a population weighted average similar to what you would see in fast exchange for static, where the limiting case is the chemical shift difference rather than the relaxation rates. You should see a linear dependence, right? And basically the slope of this linear dependence would give you the difference between the free and the bound. And of course, at zero fraction bound is the effective relaxation rate of your free state. Anyway, so that you can use this is just a curiosity, you can plot the free and bound states. Realize the way this experiment is done, this is not a pure R2, it's not a pure spin-spin relaxation rate. And that is because the way this experiment is done when you do a CPMG is you average between the in-phase and anti-phase terms. And the reason why you do that is for very long delays, because there is a J coupling between we're looking at amides, because there is a J coupling, you would see oscillation between the in-phase and anti-phase terms. Now, if you have a per deuterated product, so the difference in relaxation rates of the in-phase and anti-phase terms is so if the in-phase is NX and the anti-phase is 2NX8Z, that anti-phase term is also relaxed by the homonuclear proton T2, which for a large molecule samples the zero spectral density. The spectral density function at zero frequency, so it can be, and of course it depends on the proton-proton dipolar coupling, so it can be quite large for an undeuteurated protein. This is deuterated protein, so they would be somewhat similar, but they would not exactly be the same. Anyway, that's a curiosity. Okay, so this information can be used to define ambiguous distance constraints. And as shown here, we have some mutation data on the kinase side, and we can use the spectral perturbations I described earlier as as active and passive residues. I mean, I'm not going to read this out, but you can define these active and passive residues. And on the phosphatase size, we basically used these transient chemical shift changes as to define the active and passive residues. And these can actually be converted into an effective distance constraint. And the way it is done is and this has nothing to do with the NOE, actually. This is done because it sort of reflects the sixth part of the 612 Lennard-Jones potential, but it's a reasonable way to look at convert distances to obtain any sort of ambiguous distance constraints. And these are averaged. So this quantity is effectively a distance, and this for residue I gives you a distance between the species A and species B. And this is averaged over all the passive, the active residues of species A, and all the active and passive residues of species B. And we can discuss this more during the question hour. Okay, so this is fine. I mean, this is ambiguous distance restraints. You're not going to really using a bunch of ambiguous distance restraints where you don't know what the two ends are exactly, right? Two interacting atoms are exactly. You will not really get it's not going to be a well-defined computational problem, and so you're not really going to come up with something which is a tight cluster. And so then what we do is now we want to get some exact distance constraints, or as exact as one gets in NMR. As I say, NMR is a for structure determination, it's a complicated problem in the sense that, at least in solution, there are two issues. The first issue being you do not have a 1:1 linear transform between your experimental data and your positional, right? Like you do in crystallography. I mean, these two are related by a simple linear transform, the Fourier transform. And the second thing is that there are many complicating issues which arise because of this lack of this 1:1 mapping between your data and your atomic positions. So it is what it is. We've been dealing with this sort of thing for many, since we did structures by NMR. So we're going to continue doing that. Okay, so this is the problem here. Now the goal is we're going to spin label WZC, which is the kinase, right? Because we know where the interactions are, we know what pieces are important for the interaction, and we're going to measure the effects of spin label WZC on the phosphatase, so the kinase is going to be spin labeled, the phosphatase will not be spin labeled. So, but it will be labeled with N15, methyls, ILV methyls, and we're going to look at the paramagnetic relaxation on induced by spin labeled WZC on WZB. Okay, now there are multiple problems here. The first problem is, as I mentioned, if you add stoichiometric amounts of WZC to WZB, you basically broaden a face out. So again, that's a problem because you don't quite know what is getting broadened because of the spin label. So you have to work at substoichiometric concentrations of the spin labels, right? The other problem is WZC has multiple, actually two native cysteines which are solvent exposed. Now, if you have both of them present and if you spin label them, you have a problem because now you don't know what is what anymore, right? And because you have to work with substoichiometric concentrations to actually focus on the spin labels, again, as I said, stoichiometry broadens things out on its own without having the spin labels in there. So you have to work at substoichiometric concentrations. So you have to get pretty close to the binding site. We know these are important ones, but you can't get very close to them. because if you get very close to them, you're going to perturb the binding, right? So, and lastly, you have to get enough of them so that you get non-overlapping distance constraints between one and the other, so that you're not, you get a broader set of distance constraints, which are essentially not going to give you independent pieces of information. So firstly, what you have to do is so you're going to make. So the way we're going to go about this is you're going to take out the native cysteines from WZC, and then we're going to specifically introduce single cysteine one at a time, which then we're going to then use for spin labeling. Okay, so first of all, you assess whether this individual mutations make a difference. The first thing you see is if they are folded, right? If you make a mutant, a single mutant might unfold, these systems. Kinases especially are notorious of being, very finicky to people, probing them in any sort of way. Anyway, so, first thing you see, you make the mutants, you see that it's folded, and then you assess whether the mutations cause large structural changes. And you don't see, if you exclude the mutation sites, in this case, we looked at things which are within, we excluded residues within 6 angstroms mutation sites. And you don't see very large changes. You do see some changes, but you don't see very large changes. And these are essentially acceptable. This one shows some changes, but the next test would be to assess whether the interactions are maintained. So the native interactions are maintained, and I'll come to that in just a bit. And when we looked at the native interactions, what we found was, we prefer to use the methyl groups in this case, the ILV methyl groups on WZB to assess their native interactions. One of the interesting things we noticed that in all cases, including wild type, when you add WZB to this kinase, these are you end up seeing slow exchange. One of the things to remember, right? And this is an aside. This is not. When we discuss slow, fast, intermediate exchange, when you look at HSQC or TROSY type experiments, the chemical shift evolution, the regime is single quantum. And these were done with HMQC type experiments, so the regime is different, right? So the regime for multiple quantum evolution and single quantum evolution, which defines slow and fast exchange, may not be the same given a certain regime. Okay, and this we can discuss as we come and of course these are methyls, so these are narrower, so you get better resolution. Anyway, all of them basically showed similar sort of features, except this when we spin labeled at leucine 505, which moved from slow to fast exchange. So basically you saw gradual shifting of peaks from the free to the bound. In this case, you saw one going down, the other going up, which is characteristic of slow exchange. Though the pattern of perturbations were pretty much the same. There was essentially no difference. So basically the binding mode is not altered, but probably the binding affinity is altered in this case, making it go from the slow to the fast exchange mode. And this is actually confirmed by analysis. And the way we analyze this, if you remember, the heat release was low, so we decided to switch to NMR. And there's a very robust methodology which was developed by the Wadby group, published in Scientific Report, which uses a pretty comprehensive line shape analysis and taking into account the fact that whether you're looking at single quantum or multiple quantum. Multiple quantum means 0 and double in this case. And then you can do line shape analysis and you can sort of get, KD values. And of course, we used our, whether we get the correct KD values using wild type. If you So that's essentially what we get by other means. So these we believe, and this one, as you can see, leucine 505 shows a very, it sort of weakens the interaction because it's very, very close to the key interaction interface. So this was not used. So we just use distance constraints from these four. Okay. So then next you want to check whether, when you do spin labeling, whether it's actually, does actually label. And in this case, we didn't use MTSSL, and for the simple reason, WZB, and we wanted to also functionally characterize the system ultimately. And so we did not want to perturb the native cysteines in WZB, because it is a cysteine-dependent phosphatase, and the functional residue is a cysteine. So we didn't really want to mess with that. So we had to work with, in the presence of PSEP or DTT or some kind of a reducing agent. If we used MTSSL, what would happen is we would break, we would dissociate the spin label, and so we use this this IPSL, which actually it's permanent, so it just doesn't come off. Of course, it has lesser degrees of freedom, which may be problematic. Anyway, so we ended up seeing spin labeling. We tested it by mass spec, so spin labeling is correct. Next, we actually checked whether a lot of these spin labeling species are hydrophobic, and kinases sort of like hydrophobic molecules. And so they would get into nooks and crannies, and then we don't know whether, we are seeing effects because a piece of that labeling, unreacted labeling molecule is stuck somewhere, and that's giving us these sorts of, PREs. But clearly, so we saw that the PREs are located on one face of the, so there is no real non-specific binding. Next, we reduce the spin labels to compare it with the bound and free, and see if the nature of the perturbations are similar. Overall, they are. So introduction of the spin label really doesn't, and so this is the reduced spin label. We looked at straight spectral perturbations, compared them to wild type, just to see if the binding mode is similar, and it's not changed. So when you do this sort of thing, you have to have multiple layers of quality control to make sure that you're not getting you're getting what you think you're getting and not something else. Okay, and so of course the four different spin labels we're using essentially give you non-redundant sets of PREs. One of the things to remember, which I should have mentioned here, I should have had a slide, but of course I forgot to do it. One of the things you should remember, that when you're using a spin label species or any kind of paramagnetic species, and the PREs here were calculated from resonance intensities of the amide resonances from a Trosi type experiment. Now realize your spin label affects your T1, and T2, it affects your T2, which is what we're using. It also affects your T1, right? So when you're comparing the paramagnetic and diamagnetic resonances, do not forget to make sure that you have enough of a pre-delay or enough dummy scans, because what would happen if you don't have enough of a delay in your diamagnetic experiment, you would reach a very different steady state between scans than you would in a paramagnetic experiment, simply because your T1s have been reduced in the paramagnetic experiment. So that's something to remember if you're going to quantify things. Get reasonably good quantification of the effects of the paramagnetic effects, since you're looking at line intensities, or is essentially what we did. All right, so basically we included these sets of ambiguous and unambiguous distance restraints, and then we ended up getting Haddock. just as a quality control, we ended up doing, you use only the ambiguous restraints, you use only the unambiguous restraints, and then you use both, which is your data set, just to do some level of quality control to get a sense of whether you get consistent results. And we did. And one of the good things is, one of the things one looks at is this HADOC score, which is a reasonably good score. And one also looks at good clustering, right? And I will discuss this a little later. So so this is the dominant cluster, right? And this basically shows convergence. So if you have convergence, you would get a dominant cluster where most of your calculated structures would be. And then you get a decent buried surface area. Okay, so basically, if you look at the first cluster, you can then define what the interface is, right? And then you can identify a set of interactions, right? And as we said, clearly, these residues, these are highly conserved in the kinase, take part in interactions, and there are others, right? Because our goal ultimately is not solving the structure or getting the interaction profile is not the final goal. The final goal is to understand what is important, because then we can perturb it and then get have a biological handle on what's going on. So one can also look at the various clusters, and this is another way to look at convergence. Basically, you look at the perturbation. So you take all the structures in the various clusters and do a statistical analysis based on what the interfacial residues are, where we define an interfacial residue as something between 4 angstroms of an interaction partner. And then you basically see the major clusters. These are the 2 major clusters, essentially going up to 80% of what you have. You see the interaction interfaces are very, very similar, and so that tells you that you have it right. Now, we're still not done, as I said. We're still not done simply because, if you remember, we have now defined the interface, but we took the actual substrate out from the kinase, the substrate being the tail, which has all the phosphorylated tyrosine residues. So we have to model that back in, see if this sort of conformational mode, this interaction mode, is compatible with dephosphorylation. In other words, now if we put the tail back on, whether it can access the active site, and we can go and do that. And so we use modeler to model the tail back on, and we define two key sets of distances between the catalytic cysteine on the phosphatase and this arginine. So this is a conserved motif on the cysteine dependent phosphatases. So, and then we took a crystal structure of a mimic and see if the various tyrosines, there are 6 of them, whether they can actually access the active site in any sort of optimal way, so that they could be dephosphorylated. And all of them could except this one, tyrosine 705. And tyrosine 705, interestingly, in cells is not autophosphorylated, right? So there would be no need for it to get dephosphorylated. These ones are phosphorylated, and so these ones can be dephosphorylated. So our model is reasonable. So the last thing we wanted to do is to actually test this interface. And so I said there are 2 interfaces, and one would actually test it biologically in a sense. So we mutated these, and we found that indeed the dephosphorylation rates are basically compromised in the mutants. So we think that we have the mutation, we have the interface correct. And there are fundamental biological consequences of using this mutation, which I will not get into, but this work was actually done. So Iman and Kaushik had, Denise, many years ago in my lab had started this work with Kaushik, who's in the audience. And then Kaushik followed it up a little bit with Iman. And after Iman left, a postdoc, Sebastian, basically did most of the follow-up work. And this was published in PNAS earlier this year. But if those of you who are interested, this is a very interesting kinase system, which is unlike any others you would see in any of life's domains. So if you're interested, and we kind of know how it works now. Using a variety of techniques, we've come to this answer, and we've described this in this JBC review, which is which is going to come out in the next few months or so, and it's online. And those of you who are interested can actually go and look at the biology if you're interested. And I'm happy to take any questions, and that's pretty much it for me at this point.
Thank you, Rani. We'll have some questions at this point of time before we move on to the next speaker. So there's a lot of information we got. To start up the question session, you might have told, how big is this kinase you're looking
so you're looking at a complex. So the phosphatase is about 16 to 17 kilodaltons. The kinase is about 35 kilodaltons. So you're looking at a reasonably large, by NMR standards, complex.
And the nitrogen labeling, when we look at those HSQCs, you have uniformly labeled nitrogen 15 there, isn't it?
So
you're looking at a 35 kilodalton molecule?
That's correct. Yeah.
That's impressive. That's a beautiful spectrum there.
I mean, when you have and this is something which is interesting. I mean, this is one of the smaller systems we've studied. One of the big advantages of having this is one of these big advantages which there have been multiple breakthroughs in NMR over the years, and one of the biggest breakthroughs, I think, in my opinion, in the last 15 years or so, is the ability to methyl label these systems. For large systems, things which are above 40 kilodalton, and even we've looked at 100 kilodalton systems. If you have a structure available, you can use to a very large extent HMQC, nosy HMQC type experiments based on the structure using the methyl nosies to assign this, at least the methyl groups. In this case, and something which we worked on, which is 80 kilodaltons, we could get sufficient number of backbone assignments, and this one is essentially completely assigned, and Kaushik was involved in doing those. But having structures and having the ability to methyl label, most of the systems we study now are very large complexes, because we try to look at intact systems with their partners, and so we don't really use backbone anymore. We use methyl groups, and when we have structures, it makes life a little bit easier in assigning things.
That's impressive. The thing is, actually, how much the methodology has been pushed to study along with a lot of biology. That's that is pushing the molecular weight limit to this extent, and you are able to study complexes. That's very nice. If there are any other questions, I will probably we can come back to people may be thinking about what to ask, so we can always come back to the questions later also on your talk.
I have a question, if I could chime in real quick.
Please.
All right. So two quick questions. The first one, I really liked your protein structure images and your cartoons of the interactions. What software did you use to generate those?
So I use Chimera X because we do a lot of my people, the people in my lab use PyMol.
Okay.
Right. So unfortunately, if you're in my group, and if I'm going to make the figures, I've convinced them to learn both because, I'm not that smart and I don't want to learn a second software. But yeah, I use Chimera X
Okay.
I use that to make figures, just simply because it gives you a lot more flexibility than, at least in my hands, bimol, because we are moving towards very large complexes and
Right.
EM.
Right, that makes sense. So my other question is, you had mentioned in the beginning of your talk about the confounding factor of the phosphorylated tyrosine tail. And I was just curious if in your modeling, did you have any issues with, say, like induced fit, where you needed that tail in there for it actually to form the correct complex?
You do not need the tail. The tail is, of course, the substrate, right? So
Right.
basically how these things work is all it, and this is a long story, but I'm just going to give you the short version of this. Essentially, all it does with a lot of these things is you increase the local concentration of the protein and the kinase and the substrate. And so the interaction is driven by somewhere else. So you just bring these two molecules together. The tails are flopping around and occasionally getting into the active site and getting dephosphorylated. And this is how a lot of these sort of molecules work. Another system we work on, we call this proximity-induced activity. Essentially, you just simply enhance the local concentration of one to the other. That's it.
Okay. Thank you very much. I appreciate it.
Thank you. Very good. At this point of time, I will invite our solid state NMR specialist, Professor Andy Newcook. The floor is yours, Andy.
Great. Thanks, Murali, for inviting me, and for all of you for sticking around for some intriguing biology. Today, I'm going to talk about biological solid-state NMR. First, I wanted to introduce myself just a little bit, because I'm going to talk about a lot of things. It's not one big story. It's a bunch of little stories. So I just wanted to make it clear what I normally do. So my main project in the lab is a membrane binding project, where we're studying proteins interacting with specific phospholipid. I'll show you a little data there in the section where I talk about membranes. And then, as Murali pointed out, we spend a lot of time here at Rutgers developing methods for these very fast spinning rotors. There's a lot of NMR methods, and there's a lot of mechanical methods, for lack of a better word. There's just a lot of manipulation of stuff that goes into these smaller and smaller rotors. I don't have time today to talk about the pharmaceutical stuff. We have a paper we just submitted with some people over at NIST about monoclonal antibodies. Actually kind of fun. Might have been a better topic for this audience, but I thought it was supposed to talk about big proteins. So, well, protein structure determination. I can't solve the structure of a mAb yet. And then finally, the last project in the lab is a bunch of ionic liquids. So that's the background. But today, what are we going to talk about? We're going to talk about why you would ever want to do solid state NMR, especially since solution NMR can apparently study hundreds of kilodalton complexes now. I'll go through a little bit about how you go about solving structures, how things have changed in the last few years, what we're up to these days. One of the things I've often find with audiences that aren't familiar with some of the more recent literature on solid state NMR is they're a little surprised it looks as good on proteins as it now does, because well, solid state NMR for years was, big amorphous material type things, really broad lines. So we've come a long way from there. And then, like I said, I'll have some conversations about practical concerns. If you find yourself wanting to do solid state NMR, what do you need to do to actually do it, as opposed to the fundamental spin physics. All right, so why do we do solid state NMR? We study solid state NMR on things that are too big or too amorphous to do solution NMR or crystallography. So at the point where they get too large that even with the methyl labeling and deuteration, you can't study them anymore. Well, so solid state NMR, we don't count on the tumbling of a molecule in solution, which is why Murali was so surprised with how big the molecules are getting. In solid state NMR, we're doing mechanical spinning, and so it doesn't matter how big the actual object is. That's the solid part. And so good examples of this are these fibrils, and then, of course, membranes and membrane proteins. So you have these large membranes. They're too large for solution NMR. per se. And so we like to do solid state NMR on these larger systems, like I said, peripheral membrane proteins and membranes. And so again, how do we do this? We use magic angle spinning. I'm going to talk about magic angle spinning. There's a lot of static NMR out there as well, but today I'm not talking about magic angle spinning. The idea here is that in solution, things tumble in a natural correlation time just based on Newtonian motion of objects in solution, but large things don't do that. Macroscopic molecules don't do that. So instead we have to spin. We pick this angle, 54.7. It's the vertice of a cube. If you bisect a cube to a cube, it rotates X into Y into Z very quickly. So it removes the orientational dependence of the magnetic field to your solid crystal that isn't rearranging. It happens to pop up in this equation right here, which is the intensity of a dipolar coupling, which is going to come up a couple of times because we love our dipolar couplings in solid state NMR. Or basically, if you plug beta into 3 cosine squared minus 1, you get 0. So it removes those orientational dependencies. You get rid of the signals. You go from a broad static line shape, which we always joke has a lot of information, but it's hard to get to sharp lines when we spin faster. Of course, we want that information, so we spend a lot of time trying to get back later. So what sort of structures have been solved by solid state NMR? If you go back to early 2000, you have the first structures of oriented helices. The first sort of soluble protein structures were a couple of years later, but as you can see, those bundles were pretty loose. And as the years went by, the restraints got better and better, and you started to get tighter and tighter structures. How do you do that? Well, you solve structures in NMR with sort of a 5-prong attack. The first thing you have to do is isotopically label, almost always with 13C and 15N, because those are the NMR active nuclei of the appropriate atoms. We do a little bit of natural abundance work in my lab, but for the most part, for proteins, you need those labels, which is why labeling just the methyls is so powerful, as Ronnie pointed out earlier. Then we do the chemical shift assignment. So I'll spend a fair bit of time today talking about how we do that with proton detection, because it's a moderately new thing that I've spent a lot of my career working on. Then we collect restraints. Again, Ronnie spent a lot of time today already talking to you about how you do that, what sort of restraints you need, the importance of ambiguous versus unambiguous restraints. And it's that second, you're getting good unambiguous restraints that let your ambiguous restraints converge is really important. And that's because of the way we do our structure calculations, which are these guided molecular dynamics simulations. You have a very high energy, low restrained, in like a physical sense, molecular dynamics, where the NMR data is what pulls the structure together. And if your data is too ambiguous, it doesn't pull together at all. And so you need those high quality data to actually pull your structure together. And then you can spend as much time as you want refining structures. So it's something I've spent a lot of my life doing, coming up with new, nicer data restraints to get better and better, tighter structures. So yeah, restrained molecular dynamics. If you have a lot of ambiguity, you get very broad things. This was the first time it was ever done, so people were very happy. Ann McDermott and company did it a few years later. It was tighter. And then Chad Reinster, my PhD advisor, came along and we solved the structure of GB1 a whole bunch of times with a bunch of different data. And by the end, we had 7,000 distances, a bunch of angle restraints, a bunch of fit vector angles, and we nailed the backbone of GB1 down very, very firmly. So more data, better structures. What else can you do? You can improve the accuracy of those data. So if you get precise distances with actual being able to fit, which is actually nice, something else Ronnie pointed out, and I'll talk about a little in a couple of slides. If you have a specific expression you can fit your data to, you can get a very precise distance. And so here is a structure with 700 distances that has a better RMSD than this structure with 7,000 distances, and it's because these distances are very precise, which locks down the position of all these side chains, whereas if you have kind of vague distances, you end up with this sort of ambiguity in your restraints, which results in an ambiguity in your structure as well. All right. So the first example I want to talk about is the structure of alpha-synuclein fibrils, which was solved in Chad's lab a few years ago, and which I spent a lot of time along with many other of my colleagues. And so the problem with alpha-synuclein has very repetitive primary secondary structure. primary and secondary structure. And so its spectral is much less well resolved. So if you look at GB1 over here, this carbon-carbon correlation spectra has really, really well resolved peaks. So it's very easy to pick out unambiguous distances because the resonances are all resolved. If you look over here, you have this pile of lysines on top of each other in synuclein. Everything's degenerate. And so those restraints are all degenerate. And so the sequence is redundant and the distances are hard to identify. And so what do you need to do? You need to get specific, precise distances. And so that's what we did. We used this Z-filtered TDOR experiment. And actually, the nice thing about TDOR here is it's a mixed experiment. So you were able to label carbon on one molecule and nitrogen on the other. And we were able to see actual intermolecular distances, which is what was necessary to solve the structure of a fibril like this. And then the other thing you need to do, which I think Ronnie also mentioned, is you need to bring in data from other sources. You need to have more information. So one of the things that's very promising and exciting in NMR right now is that cryo-EM structures are getting very good. AlphaFold like predicted structures are getting very good. And so if you can start with something reasonable, then it's much easier to interpret your NMR data. And so that's something we're taking advantage of a lot, whereby NMR can help you with the fine details, the chemistry. You want to be sure that those side chains are actually where you think they are. And you have something like an AlphaFold structure that'll get the backbone right 99.9 times out of 1,000. But does it have the active site right necessarily? And so then you can go in and use the dynamics experiments Ronnie talked about, the precise distances, and actually nail down the details of what's happening chemistry-wise on top of a structure that was solved using a method that's easier than solid state NMR is. So anyway, we knew the fibrils were a specific width. We knew they were parallel beta sheets. And so what we did was this dipolar recoupling experiment. And so like I was saying, magic angle spinning, we average out dipolar couplings. So you spin at a magic angle, dipolar couplings over a rotor period go to 0. But the cool thing about that is that you can selectively reintroduce them in the indirect dimension of a multidimensional experiment, and you actually get that information back while still maintaining the sharp lines when you detect. So that's what's happening here. This is an HC cross polarization with some, maybe let's turn my laser pointer on here so it's actually easier to see. A rotor synchronized pi pulse train to reintroduce that nitrogen carbon dipolar coupling. You label that nitrogen frequency in a mixed state. You come back to carbon to detect. And so these are all carbon detected data, like I said at the beginning, and you get these nice nitrogen carbon distances. And in the fully labeled material, it looks something like this. When you dilute it so that only half the objects have the appropriate label, sensitivity goes down, but you're able to see distances that are unambiguously between two different atoms, which is what you need to solve the structure of a complex like this. And it was what let us solve the structure of these synuclein fibrils. And like I said, Marcus Tuttle, who was the student who followed after me in Chad's lab, was the person who finally put all this information together to get this final Greek key. In the end, the empirical Talos was correct. There's these kinks that we were very. This one in particular was not what we were expecting going in. And so the take-home message from all of this that's bigger than any one biological system is to trust your data, even when your data is telling you things that you don't expect. But in the end, something like 400 distance restraints were what we needed to solve this. But the key was that there was 100 of them that were intermolecular, the good ones, the ones that actually got you what you needed to put together a structure like this. All right. So that was carbon detection. As I'm sure most of you know, proton detection is the more sensitive way of doing NMR. And for years, we were doing carbon detection in solid state because we weren't able to spin quickly enough to get good looking proton spectra. So I showed you earlier this picture going up to 40 kilohertz. You can see you're beginning to see some proton resolution. If we go faster and faster, 100 kilohertz, you can actually see a lot better resolution. The sensitivity of your experiment scales with gamma to the 3/2, but so too also does our friend the dipolar coupling. So the things we're attempting to spin out with magic angle spinning also get stronger as the intensity of the gyromagnetic ratio gets higher. And so in order to get good looking carbon spectra with a gyromagnetic ratio of 1/4, protons, you need to be spinning much, much slower than you need to get protons to look nice. And so the other problem we're having with protons is they're much closer together. And so there's a lot of solutions that involve diluting the proton bath, which is, again, something Ronnie talked about earlier. When you get to these larger and larger systems, you go down to just having your methyl groups. That's keeps the interactions as weak as possible because those protons are further and further apart. We do the same thing in solid state NMR by deuterating our protein and getting rid of all of our protons and only coming back to the amide protons. The problem, of course, then is the NMR nuclei you wanted to detect have disappeared. They're not there anymore. So there's a balancing act, and I'll talk in my last few slides about some of the trade-offs between those things. The faster we can spin, the less we have to dilute. So how do we go about doing backbone assignments? So backbone assignments with proton detection are done in the same way solution NMR does it now. And it's interesting. So in small molecule NMR, the goal is to identify the primary structure of the molecule, right? You want to know, how are my atoms connected to each other? In a protein, you know the primary sequence already. You know how the atoms are connected. What you want to know is the secondary and tertiary structure. And so in order to do that, you need long-range distances. But the challenge then is to literally just identify which signal goes to which atom, because the repeating units are the same over and over again. You can't use the sort of chemistry you use to identify small molecule NMR. You have to use something else, and what we use are these backbone walks, where basically the idea is each basic unit of a protein is an amino acid. It's got an amide nitrogen, a carboxylic, a CO, and a CA. And so the idea is to a series of experiments that take this amide proton NH2D and connect it to the CO and the CA. And so then there's a series of experiments that go forwards and backwards. You have the CaNH that does the I residue, and then you have the CaCONH that comes the other direction to the I-1 residue. And the idea is that with a set of all these experiments, doing the CAs, the COs, and then not pictured here, the C betas, you can actually identify in 3 separate spots neighboring residues, which due to secondary structure differences will not all be overlapping. So different amino acids and different secondary structures have slightly different alpha, beta, and CO chemical shifts. And so as a result, having all 3 of those will let you do a backbone walk, and you actually walk through the protein. Because it's an NMR audience, I get to show pulse sequences. I'll show you another one of these. There was 2 kind of, I don't know, competing is the wrong word, different successful strategies for this. One that was developed by Chad while I was there. Another that was being developed in Lyon by Guido Pintacuda and company, Lyndon Emsley. Where basically in both cases you were starting on the proton and moving to the carbons, and then we were doing some sort of carbon-carbon mixing. The Lyon folks were mostly spinning at 60 kilohertz and doing J-based mixing here. We were spinning at 40 kilohertz and mostly doing dipolar-based mixing. The difference between those 2 is actually quite interesting, and I'll talk a little more about it later. Depending on your system, one of those might work better than the others, and it isn't always just how fast you're spinning. It sometimes depends on the dynamics within your system. You need really long T2s, and like in solution NMR to do J-based transfers, solution NMR does all these transfers with scalar-based transfers, whereas the dipolar transfers work a little bit better when things are not as ideal, which as we push into more and more real quote-unquote systems, we actually spend a lot of time in the not ideal case. Anyway, then we suppress our water. It's apparently going to have a good talk about that in a couple of months. In the solid state, we use a combination of presat and a homospoil if we have it. And then you come back to your protons and detect your residues. The other thing you can do as you spin faster and no longer have to deuterate is actually have a set of experiments that do the same thing, but go through the H alpha instead. And so that has the advantage that the H alpha and C alpha have a much broader chemical shift range than the N and the H do. Amide protons are notoriously all right on top of each other. And the H alphas are a little bit more spread out. And it also has, it's just a separate strategy, a separate amino acid. It's just another chance for things not to be degenerate. So Murali pointed out that as we've spun faster and faster, our spectra are looking more and more like solution NMR, but they're still not quite there. Okay, so the slide I was heading forward to is much further into the talk, so I won't get there. So we still have a lot of ambiguity problems just in our chemical shifts. Our line widths are something like 10 times broader than solution NMR still. And so having an extra place where you can get resolution can also be very helpful. You do interaction tracking. Again, Ronnie spent a lot of time talking about how this is done in solution NMR, same idea. Basically, you watch the chemical shifts of all of your peaks. And when you add a small molecule, you add a protein binding partner, you watch what changed, and you're able to map that on the solution, on the structure. I don't need to go into that because Ronnie showed you a billion examples of that earlier today. All right, so the next structure I want to tell you about is this membrane protein. Integral membrane proteins are a classic solid state NMR target because if in order to have a realistic looking membrane, it is very difficult to do anything but solid state NMR. Solution NMR ends up with these very small model membranes, which can sometimes cause challenging data interpretation. And so E. coli, outer membrane protein G is 280 residues. It's transmembrane beta barrel. So kind of an easy case, I would say. We've reconstituted it here in native like lipids. And the question going in was that solution and solid state and X-ray structures were different. So basically in the PDB, there were two existing structures. One was a crystal structure, which showed the beta barrel extending much further than you would expect out of the membrane. The crystal structure was solved without membranes. But it did show this interesting pH-dependent gating mechanism, where basically, if the pH, if this histidine was protonated, the structure was different, and so there was an observed pH-dependent of this structure of this channel. So everyone thought, "Yay, it worked out." The structural biology told us the right answer. A year later, a solution NMR structure came out with a substantially shorter beta barrel that better matched the width of the actual expected bilayer, but with none of this gating behavior. And then there was concerns from the crystallographers that the model membrane they used for the solution NMR was too short, it was too detergent-like. And so therefore, the reason this beta barrel didn't look as structured is because the solution NMR was done with bad lipids. And so therefore, obviously, you couldn't trust the solution NMR. So solid state NMR needed to come in and solve the conundrum. You will note, if you were paying attention, that the crystal structure is from 2006, the solution NMR structure is from 2007, and we came to the rescue only 10 years later with the answer by solid state NMR. So solid state NMR still needs to work a little bit on our pace of answering questions before people will take us 100% seriously, but we are making a lot of progress in that regard as well. So we use those experiments I was just telling you about, those cross-polarization experiments, to assign all of the residues in the beta barrel, and amusingly, very few of the residues in the loops. Solution NMR was able to see one of the loops, but not the one with the histidine that was so important. So this was the problem, right? The histidine part that was supposed to be the gate wasn't showing up the solution NMR. But interestingly, what this is demonstrating is that the rigid parts of the protein are in the bilayer, and then the loops parts are just much more mobile. It's kind of the exact opposite of what you see in solution NMR. So it actually often makes the two techniques very complementary, where the rigid parts don't show up in the solution NMR and do show up in the solid state, and kind of vice versa. The structure was solved. I took out most of the slides with the details because I didn't want the talk to go horribly long. But we had 240 proton restraints, of which 100 were unique. But really, the key to getting the structural bundle to come together were these 1,000 carbon-carbon restraints. But the problem was they were very ambiguous until the proton-proton restraints, especially these 57 long-range ones, which were able to zipper up the beta sheets, came along. And so basically what we had to do is we had to use ambiguous carbon-carbon restraints, ambiguous restraints like Ronnie talked about, but using algorithm that allowed us to add in the unambiguous proton restraints first, get a kind of original fold, and then add the carbon data in afterwards, which allowed us to get a good structure by using the ambiguous data as much as possible. Whereas if you just threw the ambiguous data all in at the beginning, nothing would happen. And then some hydrogen bonds to zip up the thing. And then some biology for you. You can look at the difference between the crystal structure and NMR structure, you can see that there's actually this helix that lies on the surface of the membrane. It's actually a hypothetical, it could be a binding site for the sugar that actually comes in. And then it was confirmed that this extra long beta barrel is actually just a result of the crystal artifact. There's crystal contacts between, the crystallization conditions just form like a beta barrel of infinity. And so they get really positive interactions there that let it form a crystal, but as a side effect, the sheets go dramatically higher than they do in real life. All right, so there's an example of a structure that we can solve with proton detection that Joran Rattel, this was a work that I did with Hartmann Ashkenat at the FMP in Berlin, and Joran Rattel was the first author, the person who did most of the computational work. I was really only taking NMR for that experiment. All right, so last little mini vignette, I want to talk about our membranes. So my lab studies phosphatidylanoacetyl phosphates. It's a type of phospholipid. It's a signaling molecule. It's got a very important inositol head group that binds a bunch of binding partners. And the different populations of PIP on the different membranes are sort of signposts throughout the cell. And so the biggest problem we had when we started this project was trying to get, we call it solid state NMR, but most of the samples we study aren't solid in any appreciable sense of the word. They're very squishy. Squishy state NMR is maybe not as popular of a statement, so we call it solid state still. But these liposome pellets are extremely loose. So after a couple hours in the ultracentrifuge, you get a very not solid object, and the goal is to move that, which could have thousands of dollars worth of material in it, into a rotor that's about the size of a dime here, without losing your sample. And so the strategy here is to take the ultracentrifuge tube, place it into a machined funnel, put that whole thing into a 50 mil Falcon tube and spin it into the rotor. The liposome pellet goes straight in. There's no manual interaction with the pellet. If you start poking these pellets with like spatulas, they fall apart. It's really touchy. So that's what we were doing with the 1.6s. We then moved on to the 0.7s. And the 0.7s, if you've not seen one, are 0.7 millimeters in diameter. They are very, very small rotors. So what I didn't talk about when I talked about MAS at the beginning of the talk is we're spinning our samples pneumatically with air. If you think about the ways one normally spins things, alternating electrical currents are not your friend inside an NMR magnet. Nor can you spin metal inside of an NMR magnet. And so what we have to do is use air. And so the rate limiting factor on how fast you can spin these rotors is the speed of sound on the surface of the rotor. And so to spin faster, we get smaller or we use less dense gases. And so the alternative is to spinning in air is to spin with helium, but that gets very expensive very quickly. So the kind of leading technique in the field at the moment, the predominant, maybe is the better word, is to use gas or nitrogen, air or nitrogen. And so to get spin faster, we just have to get smaller. And so to get smaller, you have to build smaller and smaller tools. And one of the we were running into, this tool was built for us by the machine shop using traditional wave type techniques. But for these smaller and smaller rotors, the tolerances were getting to the point where it was really nice to be able to do something in-house. So my postdoc, Tom Osborne-Pop, got a 3D printer with his fellowship. And so we started 3D printing these, and we were able to try a lot of things over and over again before we got something that would do the same thing that lets you spin a sample into a rotor. And in a similar problem, we ran into with the proteins. So I was showing you GB1 spectrum earlier, and you get these really lovely solution-like spectra of GB1. But the problem we were running into is we weren't always getting spectra that looked like this. We were sometimes getting spectra that looked like this, which are kind of okay, but not great. And then we were getting spectra that looked like this, which are just kind of not good at all. And then we were getting spectra that looked like this, that are just clearly terrible. And this rotor was almost entirely water. But the problem is that just anything kind of homemade to get protein into a rotor this size wasn't working great. And so we developed a tool that was twofold. The first was directly transfers from either an ultracentrifuge tube or an Eppendorf tube into our rotor. Amusingly, we have yet to find a material better for transferring than the pipette tips we've been using in sort of home built for a long time. For a while, I felt bad about that. I was like, why are we using pipette tips? That's like janky and homemade. But then it kind of occurred to me that these things have been engineered for, I don't know how many years, decades at least, to be really low retention, very smooth, good transfers. Like the whole purpose in life is to do this. So why reinvent the wheel? We basically built our 3D packing tool around a pipette tip. And this will go directly into the ultracentrifuge. You put your liposome up here, it spins it directly into the rotor. And then we transfer this material in a humidity controlled chamber to keep our water from baking off. And so the idea here with this is that, we maybe got 4 out of 16 rotors right, and we're 2 for 2 so far with GB1 since. So N is not huge, but these rotors cost a lot of money, and once you get a good one, you tend not to pack a bunch more. So yeah, if you find yourself asking, wanting to pack any of these types of rotors, do let us know. We have made 1.3s and 1.9s and 1.6s and 0.7s for a variety of people. We're having, people report back to us on what works and what doesn't work. So we're trying to be very iterative about this. Nobody wants their samples ruined. So definitely reach out if you are interested in packing a rotor size. All right, so changing gears a little bit, or continuing the story about liposomes, what do we do with our lipids? Lipids are fun because naturally occurring. I was telling you earlier, for our proteins, we're constantly having to isotopically label with carbon 13 and N15. For the lipids, we're able to use the protons now that we're spinning fast, and phosphorus 31, both of which are 100% abundant, naturally speaking. And you can get nice looking proton and phosphorus spectra of our lipids, which give you handles to do measurements. So Ronnie was showing you a bunch of T1, T2 measurements, things that will tell you good things about your samples. So here we're using T2 to look at PIPs in membranes with a variety of conditions: calcium, phosphatidylserine, other lipids. So you're able to study the dynamics and sort of the structural organizations of these lipids using natural abundance isotopes with solid state NMR, and actually look at the differences in the T1s of the head group phosphates under a bunch of different conditions. We're actually working really hard right as I speak on a bunch of different calcium concentrations and seeing very strong trends on the head group T1s. Actually give us indications of what calcium is doing as a signaling molecule in this system. Same story with terms of assignments. Here's something you'd be very familiar with, an HC het core in small molecule. And then I wanted to show this because I always find it interesting. The blue is the solution NMR experiment, the red's the solid state NMR. The blue is proton detected, the red is carbon detected, and you can see, as is often the case with these, you almost get little X's. The carbon line widths on the indirect dimensions of a solution NMR experiment are sampling limited, so they're broader than the solid state experiment, which is carbon detected and therefore not sampling limited. And in reverse is true. We have broader proton line widths in the proton spectra, and so you end up with these little T's, which always fun. But the overall goal is to take solution assignments in a sort of individual small molecule sense. In this case, the lipid was in chloroform, and then transfer it over to our assignments of the lipid in a actual lipid bilayer, which is the position we're most interested in for the biology. All right, so how long longer? I got a little bit more time. Like I was telling you about earlier, one of the things we've been spending a lot of time in solid state NMR is thinking about what size rotor we want to use. And so, as you know, NMR is very sensitivity limited in the most part. And so as we were wanting to spin faster and faster and faster and end up with less and less of our sample having to be deuterated, get more and more protons, we are also getting rid of a lot of material. And so when I was a PhD student, we were having interesting science about the difference between 20 and 10 milligrams going from a 3.2 to a 1.6/1.9. So you got twice as much spinning speed with half as much material. And so kind of intuitively, that felt like it would maybe be okay, because it was a factor of 2 in both directions. The very next leap, though, was a factor of 5. So 5 times less material, and you were only getting 50% faster spinning. And this got a lot of people worried that, yeah, you're getting sharp lines, but like if you have one nuclei in atmosphere, in a vacuum, it would have perfectly sharp lines, but you wouldn't be able to see it because you don't have any sensitivity. So at what point are we starting to break off that trend? And then again, with the 0.7s, 0.4 milligrams, 100 kilohertz. And if you were at ANC this year, they showed the first 0.4s with 200 and some odd kilohertz of spinning. And for that portion, it was very clear that you have reached the spot where even with sharper proton lines, we're starting to lose on sensitivity overall in the first dimension, which is, I think, the really key point of this whole little conversation I'm going to have here. And so I don't want to belabor this data. It's a little complicated, especially since there's a lot of 60% and 40s and 60s floating around. The blue is 60 kilohertz at 100% back exchange. And you can see for the NH2D fingerprint spectrum, you're losing a factor of maybe 50% in sensitivity, which you sort of expect because you're at only have 20% of the same sample. But that being said, you've already made up something like a factor of 3 of that factor of 5 just because of the sharper line widths. But then the real take home from this whole very exhaustive, moderately excessive study was that the further and further out you go in your assignment spectra, the more and more of those transfers you need to do. So the experiment I was showing you earlier was one of these CBCA-NH or the CaCO-NH. So there's a HC transfer, a CC transfer, a CN transfer, and an NH transfer. Each of those is not 100% efficient. The faster spinning had better T2s, had longer T1 rows, and had nicer transfers. So transfer efficiency went up. And so if you spend too much time over here thinking just about the efficiency of the easiest to do experiment, instead of over here thinking about the efficiency of the harder to do experiment, this experiment takes an hour, this experiment takes 3 weeks. If this experiment is twice as strong as this one, it takes 1 week. If this experiment is twice as long, you save 5 minutes, right? And so as we look at smaller and smaller rotors spinning faster and faster, I just want everyone to remember that it's not the easy experiment we are trying to get with these fancy new rotors. It's the hard experiment, because the hard's the one you spend all your time anyway. Yeah, that's what I have to say. Oh, one more thing there. As we start to study membrane proteins and real proteins, the argument of how much sensitivity you can get in each rotor is entirely predicated on the rotor being full. And so when you show up to a biologist and say, hey, I love your membrane protein, I'm glad you can express it. Can you express 25 milligrams of triply labeled membrane protein for me? They either go into anaphylactic shock, or they laugh at you, or they turn and run away. So if you only have a milligram of sample, put it in the smallest rotor you can find, because you get the most sensitivity per mass unit with the fastest spinning. So yeah, 100 kilohertz. This was something that was spearheaded in my lab by Jackie, where we were able to show, along with many others, that 100 kilohertz, you don't need to deuterate the side chains in order to get nice amide backbone spectra. So the blue here is 100 kilohertz of GB1. The red is 40 kilohertz of deuterated GB1. And you just get nice equivalent lines without having to deuterate, which is important for samples which are hard to express in a deuterated fashion. Show you a little bit of the real project that's going on in my lab. We're studying a peripheral membrane protein, Kindlin 2. I showed it on the very, very first slide without any introduction. But the overall idea of the project is that we have solution NMR of the soluble protein domain. So you can see that in blue. When I say our solid state NMR is not quite there on real samples, this is a good example. The solid state NMR in red there at 100 kilohertz gives reasonable-ish lines, but when you compare them directly with the solution NMR like that, you can tell we have a little ways to go. But the nice thing is, because the assignment spectra are the same type of experiments, we can take the solution NMR assignments and do the backbone walk and the solid state data. And so anybody who can tell you that you can do that pretty backbone walk I was showing you earlier on this data is trying to sell you something, but because we had this solution data to compare it with, you're able to make those walks comfortably, because you're not trying to be 100% certain. You're just trying to follow the differences between your solution sample and your solid state sample. So here's the binding to the substrate in solution, and here's the binding to the substrate on a surface of a membrane. We were working hard lately to try to make that blue spectrum look a little bit better so that we can publish this for you. It's still that's still a little broad even for our tastes. But the idea here is that do the easy part in solution NMR when you can, and then when you have to do the very hard solid state NMR on the membrane in the actual biologically relevant system, have as much information before you get started as possible. All right, so the work I showed today, some of it was done in my lab. Tom, I told you about with the rotor packing devices. Rashawn and Brandon helped with those. Robert does a bunch of simulations I didn't talk about today. Ashley does the lipids. Jackie did the 100 kilohertz stuff. The synuclein structure was done by Chad Reinster's lab at the University of Illinois, and the OncG structure was done by Joran Ertel and company with Hartmut at the FMP. And of course, happy to answer any questions you might have about all of that.
Thanks, Sandy. One of the things I was thinking that probably as you go smaller in your rotor diameter, one of the advantages that you are also using shorter pulse widths and less power.
Yep, the Qs go up, the power on the coil gets higher. We're able to use more decoupling. So one of the things that's been very clear, even for carbon detected work with the 1.6 probe we use, very comfortable on a 1.6 with 150 kilohertz proton decoupling, which on a 3.2, you tell your facility manager that you are decoupling 150 kilohertz, they're like, okay, no more enema time for you. The spectrometer is booked. So yes, I pointed out the sharper line widths, but the higher Qs and the more sensitivity just from the coils are smaller. Everything's nicer. Except if you put too much power through and they melt. But if you don't melt a coil now and then, are you even trying to do NMR?
And also probably your average Hamiltonian theory is more accurate because you don't have pulse imperfections. And
the problem is that the rotor periods are getting shorter too.
That's true, yeah.
The dipolar recoupling I showed you is kind of fun, whereby when we were spinning at 13 or 11 kilohertz, you were using whatever, 5 microsecond nitrogen 90s. So there was something like 20 milliseconds of RF on for a 100 millisecond rotor period. But when we're spinning at 100, the rotor period's only 10 microseconds now. And we're getting awful close to some sort of weird steady state approximation where the 90s take up the entire, and you're doing something else. It's like adiabatic recoupling at that point. And so there's a lot of smart people thinking about what exactly it means to do dipolar recoupling at extremely fast spinning rates.
Let us see if there are any other questions for either speakers. We can entertain that right now.
I've got a question. Thank you very much, Andrew, for the excellent talk. And I was wondering if you could just comment on when would it be advantageous to use your solid state NMR as opposed to something like RCSA, residual chemical shift anisotropy?
Yeah. So CSA, it's never one or the other, really, right? It's always about getting as much data as possible. The bigger the system gets, the more likely I think I would want to use quote unquote real solid state NMR. Do whatever experiment you can get data on, right? The nice thing about solid state NMR as opposed to a residual CSA or residual dipolar couplings like you see in solution NMR, I guess it's two kind of different things, is you control what you're reintroducing in magic angle spinning solid state NMR. So you can get the full dipolar coupling back, or you can get the full CSA back, right? You don't need to take a residual one. You can just completely recouple. I actually don't know if I have that slide here. No, I don't. The same way you reintroduce a dipolar coupling with a train of pulses, you can introduce the CSA in the indirect dimension as well. And we do that. Line fitting gives you a lot of really cool information, right? And so you get a lot of control in solid state NMR. You have to deal with very broad lines relative to small molecule solution NMR, but which is why I always say, you know, exhaust all other options before starting to do solid state NMR. But at the end of the day, when there's nothing else left to try, solid state NMR will still be there if you want some site specific dynamics of something that's way too big to do anything else on.
All righty, thank you very much. And my other question was sort of more of a comment. I thought it was really interesting when you're talking about the smaller tube formats, and I was thinking to myself, well, you keep going smaller and smaller, how small can you go? And it seems like, well, you end up hitting other limitations, like you're saying, they're trade-off between the sensitivity and the line width, and then other parameters that go into the instrument itself. So just really interesting.
I mean, a lot of people, a lot of smart people working on this. We've been using zirconia for a very long time because it's very strong, but we're starting to get to the point with those 04s that is kind of, most of those 04 rotor is wall at this point, as opposed to the hole in the middle of it. And so people are beginning to think again, if there are other materials that were helpful. The very first rotors were stainless steel, and we've gone away from that, but you know, all these cool materials, you find something that's non-ferrous that you could spin in a magnetic field that's got a better tensile strength. I don't know, diamonds have been bandied about. Yeah, every time people think we're not going to be able to go any faster, 5 years later somebody spins a rotor a little quicker.
Very cool, thank you so much.
Thank you. Can we stop?
Okay.
Thank you.
So
I hand over to Dave, and
okay,
thanks
well,
for the speakers.
Okay, I really enjoyed the talks, and I'm really happy to see some bio NMR in our and I'll hope to help Chris find a few more workshops like this. So if there are no other questions from the audience, then I will thank everyone for being here, and I'll thank everyone for John Webb as well. And with that, I will close the meeting.
Very good. Thank
Bye,
you.
everyone.
Bye.
Okay.